Good morning, ladies and gentlemen. I am Sutishai Takawong from Medical Technology International Program, Secure of Hair Science, Balilak University. Today, I would like to present research projects about protective effects of phytoestrogen, low in deep enteric surface or DES, microemulsions on vascular endothelial dysfunction, and our advisor of this project is Dr. Kantipit Gongpon. This is the overview of this project that we will talk about for today. Today, we will begin with the introductions, objective, material, and method, result and discussions, and finally, we will talk about the conclusions of the research. To begin with the introduction, cardiovascular disease or CVDs is a condition that causes abnormality in the heart and blood vessel. This has many forms such as atherosclerosis, hypertensive heart, valvular heart disease. Cardiovascular disease is leading case of death in Thailand and abroad. In some cases, there are no symptoms of underlying disease or there are symptoms of another disease. With each case, the patient does not receive the correct treatments and can reach to death. The most common CVDs is the myocardial ischemia. Myocardial ischemia is the heart muscle not getting enough blood, which contains oxygen and nutrients to work as it should. It is occurs when blood flow to myocardium is obstructed by partial or complete blockage of coronary artery due to a build-up of atherosclerosis that decreases blood flow in coronary artery. The imbalance between myocardial oxygen consumption and delivery causes cardiomyocytes death and myocardial infarction. Restoration of blood supply to the heart tissue can minimize the myocardial damage, which is heart tissue can minimize the myocardial damage, which is called reperfusions. Myocardial ischemia reperfusions is cellular injury resulting from a period followed by restorations of blood flow to the ischemic tissue, resulting in a new damaging process, which causes the patient to have calcium overload, excessive generations of free radicals, and subsequent inflammatory reaction. Consequently, the prevalence of myocardial ischemia reperfusions is associated with sex difference and age. In general, CVDs rate in premenopause women are lower than those in men of the same age. The risk dramatically increased in women after menopause or aging. Aging women after menopause have many changes, such as reductions in bone density, decreased visual acuity, and hormone changes that are all part of aging processes. Different hormones are affected by aging in women. Women after menopause experience and decrease in sex hormone estrogen. Estrogen is an essential hormone that is most often associated with women. It's help develop and maintain both in reproductive system and females' characteristics estrogen. Estrogens contribute to cognitive health, bone health, and the functions of cardiovascular system. Therefore, estrogen deficiencies in menopause women is the major cause of the development of ischemic reperfusions injury due to the loss of cardioprotective effects of women's hormone during menopause transitions, which is associated with vascular endothelial dysfunction. Endothelial dysfunction is the most common form of vascular abnormality. Endothelial dysfunctions occur when endothelial cell cannot release substrate, leading to an ENOS, direct regulations, and decrease in nitric oxide. Bioavailability that control vascular relaxations and contractions, as well as enzymes that control blood clotting, immune function, and platelets adhesion. The decline of endothelial functions it providing for vascular complications such as atherosclerosis, hypertension, and cardiomyocytes, which are the leading cause of mortalities in ischemia reperfusion. Endothelial dysfunctions is the central anomalous pathophysiologic process. It is a primary antecedents to developments of both obstructive and non-obstructive coronary artery by generating endothelium drive essential vasodilators and signaling compound. Several studies reported that estrogen is crucial for vascular health to minimize ischemia and reperfusion. Therefore, hormone replacement therapy, or HRT, containing estrogen will most likely effective treatment for menopause women. Hormone replacement therapies is a treatment relieve symptoms of menopause with replacement hormones that are at a lower level you approach menopause. This non-genomics pathway for estrogen's actions in the cardiovascular systems is the activations of endothelial acyl nitric oxide synthase that results in the arterial vasodilatations in response to acute administrations of estrogen. In natural setting, we found the estrogens in phytoestrogen form that's occurring in plants that's what is situated in Pandorea candere or white cloud clear. Pandorea candere or white cloud clear are family Leguminosae is a Thai medicinal plant with a long traditions of medicinal consumptions among menopause women for rejuvenations and estrogen replacement is known as which sort of phytoestrogens, including isoflavonoids and the highly estrogenic myoestro and the oxymyoestro. Because of its known as phytoestrogenic constituents and plants drive compound that structurally or functionally mimics mammalian estrogen. And they have been applied to treat different forms of cancer, heart disease, menopause symptoms, and osteoporosis. And Pandorea candere is used in alternative medicine for hormone replacement therapies or ERT by using phytoestrogen that's occur in plants. Phytoestrogens are driver groups of non-steroid plants compound that can behave as estrogen and occur naturally in most plants, fruits, and vegetables. Many phytoestrogens seem to have higher affinity for estrogen receptor beta or ER. Steroid estrogen, which suggests that they may exert their action through distinctly different pathways. There are three main types of phytoestrogens: that's isoflavonoid, common stance, and lignin. In other menopause women with low level of isoflavonoid metabolism by bacteria may experience little or none benefits for isoflavonoid. Phytoestrogen. Isoflavonoids, common stance, and clones are found in the roots of Pandorea candere. The Pandorea candere, permanent Pandorea candere isoflavones, PMIS accumulate in their glycoside form, such as purines, glycines, and genistein. The bioavailabilities of glycines and genisteins is relatively low compared to of dianisines and genisteins. Nevertheless, phytoestrogen from plants have chemical structure similar to those in the aromatic ring. It's not easily delivered into the cell, leading to an idea to increase the transport system of these are composed by hydrophobic deep enteric solvents in microemulsion forms. Hydrophobic deep enteric solvents, or HDESS, are now very knowledge as a new class of ionic liquid or IR analogs. Because the physical property of NDESS are similar to those of other ionic liquid, while their chemical properties suggest application area that are significantly different in the design of green chemicals. As the ESS are considered one of the most versatile alternative media with widespread application. As the ESS contain large non-symmetric ions that have low reactive energy and hence low melting point, they are usually obtained by the complexation of quaternary ammonia salt with an amytrol salt or hydrogen-borne donor. SDES provide many important benefits, including biodegradability, low toxicity, low cost, sustainability, and eco-friendliness. And for the critical property of HDES to be beneficiously applied in this study, there is no need for the purification process after synthesis to require various polarities and high stability. Furthermore, HDES in microemulsions forms can increase the probability of compound into cell. HDES can be separated into two types, ionics and hydrophobics. Natural, the deep enteric solvents in the chloride is being studied as ionic liquid. For the objective is to determine the protective effects of phytoestrogen from polyacandary loaded in microhydrophobic deep enteric solvents on vascular anterior cells. Next is the material and methods. For the cell of these projects are endothelial cells. Polyacandary extract preparation. Polyacandary or PC's extractions was performed into three functions, including PC extract, PC dissolved in DMSO, and PC load with 0.1% micro SDESS or PC micro SDESS. Then adjust the concentrations into 50, 20, 10, 5, and 1 microgram per milliliter. Before performing experiments, the method of cell culture techniques, endothelial cell line, will be cultured in humidity 5% of carbon dioxide at 37 Celsius. Check the cell population densities under microscope every days and transfer to new media every 2 to 3 days. Subculture will be performed by using trypsin EDTA to detect the cell from the flat surface and notice under the microscope. Then transfer the cell suspensions to a new conical tube by using 0.5 to a centrifuge at 1,800 RPM for 10 minutes. Discard the spermicins above the cell platelets. Pour the cell platelets or resuspense in the new culture media, 1 milliliters, and transfer 1/3 of resuspended cell to a new flask with 8 milliliters of media. The method of cell viabilities by entity assay. The entity assay is widely used for measuring cell visibility by the colorized metric assay. In this assay, entity measure the reductions of yellow dye to the blue products formed by mitochondria, succinase, dehydrogenase. EAR-926 cell will be seeded with 8 times 10 per 3 cell per well place and incubator overnight. Culture media will be discarded at Perelia candidate extract in different concentration, 1 to 10 micromolar mass, into its spill and incubated under 37 degrees Celsius for 24, 48, 72, 96, 120 hours. After incubation time, the media will be brought out then at 100 microliters of entity solution concentration, 5 mg per milliliter, into its spill before incubate for 120 minutes. Formazole crystal will be generated in the reactions when the time reached 120 minutes. Discard the entity solutions before adding 100 microliters of DMSO to dissolve formazole crystal. Then the cell viabilities will be measured absorbance as a wavelength 550 nanometers using a microplate spectrophotometer. Absorbance value was directly proportional to the level of living cell in the tasting vial. Experimental results will show in the forms of relative cell viabilities or percentage of living cell in the tasting group compared to the negative control groups. The methods of determinations of NO secretions by Gray assay. The measure of nitric oxide under Gray's diazonization reaction, that reactions between sulfonylic acid and nitric profile diazonium salts. These products were treated with N plus N-1 new fetus and ethyl vinyl diamide dihydrochloride and give a colored compound that's measured absorption at 547 nanometers wavelength. For briefly, EAR-926 cell will be seeded at 8 times 10 power 3 cells per well in 96 plates and cultured overnight. Then the old culture media will be discarded and add a new cells culture media with the phytoestrogen loaded in microemulsions (DES) and incubated for 24, 48, 72, 96, and 120 hours after incubation time. The culture media will be transferred to the new 96 real place and added rice reagents. Then the reactions will be incubated for 30 minutes, finally measured absorbent at the wavelengths of 547 nanometers. Next is the results and discussion. For the table 1 and table 2 is the concentrations of micro SDES preparations by SDESS were prepared by mixing oils x-acid method 1 per 1 between 20 or 80 polyethylene glycol and water, as well as molar radiations by stylings and heatings at 70 Celsius for 20 minutes, which shows the unknowns number 2 to do in the further experiment. The result of cytotoxicity and endothelial drive and all secretions of peculiar candidate extraction dissolved in DMSO show the percentage of endothelial cell viabilities treated by PC-DMSO as compared with the control group. Cell viabilities were measured by entity assay, and endothelial cell death has been treated with PC-DMSO were compared with a control group as shown in Figure 3. The viabilities of EA's high 926 cell decrease dose and time dependently with the increased concentrations of PC-DMSO. Endothelial drive and all secretions were measured by GRI assay. Endothelial cell death has been treated with PC-DMSO were compared with a control group as shown in Figure 4. And all secretions of endothelial cell was treated to increase by the increasing concentrations of PC-DMSO when incubated for 20 hours and did not show any significant difference when incubated for 72 and 120 hours. The result of cytotoxic disease and endothelial drive and all secretions of peculiar candidate extract loaded in 0.1% micro HDESS show the percentage of EA high 926 cell viability treated by PC's micro HDESS as compared with the control group. Cell viabilities was measured by MTT assay. An endothelium cell that has been treated with deep PC micro HDES as we compare with a control group as shown in the Figure 5. Percent cell viabilities of endothelial cell was decreased in concentrations of precise micro SDESS when compared to control group. Endothelial drive and all secretions were measured by GRICE assay. Endothelial cell death has been treated with a PC's micro HDESS when compared with a control group as shown in the Figure 6. And all secretions of endothelial cell treated with the PC's micro HDESS did not show any significant difference when compared with a control group. And for the result of cytotoxicities and endothelial drive and all secretions of perolear candidate extract show the percentage of candidate cells, viability treated by PC's extracts as compared with the control group. So viabilities was measured by MTT assay, and the endothelial cell death has been treated with PC extract were compared with a control group as shown in the figure 7. Percent cell viabilities of endothelial cell was decreased by the increasing concentrations of PC extract, endothelial drive, and all secretions were measured by Grice assay. Endothelial cell death has been treated with PC extract were compared with control group as shown in the figure 8, and all secretions of endothelial cell treated with PC extract did not show any significant difference when compared to control group. Above the results I presented, the cytotoxicities anti-inflammatory effects of pure perolear candidate extracts has the highest percents up to viabilities and no secretions, while the results of cytotoxicities and anti-inflammatory effects of deep enteric solvent perolear candidate extracts has high presence of survivabilities in the low concentration. And for the conclusion, according to the experiment, phytoestrogen perolear candidate extract combined with hydrophobic deep enteric solvents in microemulsion forms did not display cytotoxicity and anti-inflammatory property. Consequently, they provide more research is required to determine the protective effects of phytoestrogens from the perolear candidate extract loaded in hydrophobic deep enteric solvents in microemulsion forms on vascular endothelial dysfunction. And all of this is our reference. Thank you.